you haven't been here before, you drink water or something, there's a water fountain right around the corner here. Don't feel bashful. There's a way to get back around to the back of the building, but I don't want to tell you how to go that way. You're making a must. But don't feel bashful, just go right out the door. Some cups in there. That's kind of a little kitchen area over there. If you want one and ice in the refrigerators. I don't think there's any fresh coffee, unfortunately.
Do you have water, Jared?
I have some. Thank you. Well, I'm really glad, I'm going to take it for granted that those empty seats down there are the people who read the announcement to for our meeting tonight and really did think that the meeting was taking place on February 20th of 2013. So, which I just noticed tonight. My vaunted proofreaders failed me again. No, anyway, so you all made it, so I'm real happy to see you tonight. Well, I've handed out what I hope is a bright yellow sheet to each of you. And this presentation tonight is, you know, as presentations that we've done here go, this one's kind of sciencey, you know, in the sense that there's some terminology that we're going to be using tonight that's probably not going to sound familiar. There's one right there, very first item. And there'll be several other terms that we're going to throw out. And you know, why do we use terms that are like unfamiliar? Because they express something that would just take too many words to express in language that we all understand. So it's this scientific shorthand, right? You give things odd names like this. And there'll be several more that you will come across during the course of the presentation. I think all of those are on that sheet. So you can call it a little cheat sheet if you want. On the far left there's an alphabetical column of terms. So if in the middle of this thing I use a term and you go, oh, I forgot what that was. You know, I'm not going to point you out if you click down. What is that? So they took quite a while for me to get used to, actually, and I may even mispronounce one or two of them on occasion tonight. But, so you can laugh at me if I do that. So there's that. There's also on that yellow sheet. There's some little resources for you on the. Oh, see, I left them in the back of my car. There are 3 books from RTB. That I used in preparation for this, and ones that I think you would enjoy very much. One of them, and I'll just mention one, is The Origin of Life, which is an excellent book written by Hugh Ross and Fazil Rana, who himself is a doctorate in biochemistry. And that book touches on a lot of what I'm going to be talking about tonight, plus has a lot more. We're going to talk for about slightly less than an hour, but you can read that book night after night. New stuff every night and be thrilled. The other 2 books are called The Cells Designed by Fazil Rana, and that one's actually more like a textbook. That one's pretty tough. That one was really hard for me to get through. Again, I've forgotten the name of the 3rd one, but if you're interested in looking at any of those, just ask me afterwards and I'll bring them in. Okay, I'm going to draw your attention to the very first item on your sheet there, which I can see I didn't read myself one, but I think it says amino acid. Yeah. And that's one that we're going to encounter a lot during our presentation. So again, we'll try to explain it here in just a minute, but that's just where we're going to start. Now, as I stated in the opening sentence of the announcement that we made for this presentation, the origin of life on Earth. The origin of life is a mystery. I mean, it's a deep, deep mystery. And I pause here a little bit because I really don't want you to, I don't want this simple fact, and it is a fact, nobody argues it. I don't want it to be forgotten in the many details that we're going to be covering later. Because despite what you may read in some books written at the popular level or see on the internet or even here in a biology classroom, you know, when I say nobody knows the origin of life, that doesn't mean there aren't a lot of people with a lot of ideas. There are. And sometimes those ideas get expressed with a degree of what I would call certainty that is not really there, should not be there. So I'm going to say that in scientists all across this country, and in fact the entire world, fight amongst themselves over the origin of life. And one of them will say, I have this great idea, I think it happened this way. Some other scientists will say, well, that's what you think. And I've got some research over here that says something entirely different. And let's face it, they're all fighting for research grants. So these things happen in science. I'm not afraid to cast aspersions on them. But, so even what you might read in here in a biology classroom, no theory of life, of how life began here on Earth, enjoys anything approaching a consensus. You hear people talk about a consensus on global warming. Well, whether that's true or not, really all they mean is most scientists would agree that, say, global warming is occurring. Well, most scientists, there aren't any group of scientists who would agree amongst themselves that we all know what the origin of life was. They don't. And even when you hear the various theories that people propose, those theories really enjoy, I guess, a diversity that's really almost breathtaking. And you're going to see some of that tonight, just how far apart some of the ideas can be. However, having said that, it's also important to note that there is something approaching a consensus within the scientific community, and especially in the biology community. And that is that the origin of life was a purely naturalistic process. However mysterious it might have been, however improbable it may have been, there was nothing supernatural involved. Life is simply the result of cause and effect in a closed system. That closed system being our universe. Okay, there is a consensus out there that this is true in the scientific community. And that even though we don't know all the details, the chemical pathways that led to the establishment of life will eventually be understood. We will discover those things, and all the mysteries will be solved. Now, I think it's almost a truism in science that we don't often find disagreements when it comes to raw data that people collect. Okay, I measured the temperature over here for a week, and at this time during the day, and I got these numbers. People don't often argue over that kind of thing. However, they sometimes do argue over the relevance of it. Okay, you've got this data, so what? What does that mean? But people often disagree as to the popular conclusions to be drawn from the data. I may have this temperature data over here, and it shows the temperature going up every day for a week. Oh, global warming must be true. Well, of course not. I mean, that would be silly. So we would disagree in that case about the relevance or about the conclusions to be drawn from that particular data. But case in point, reasons to believe. Okay, what is reasons to believe all about? Well, reasons, RTB, is predicated on the belief that there's a creator, and that the data obtained by scientific research supports the notion that we can see God's hand in creation, or at the very least, we can strongly infer it. And of course, many scientists out there in the world disagree with that. They don't see any need to invoke the handiwork of a creator or a supernatural being. Now, this is not a presentation on evolution. Don't have time for that. But it's important to start by asking the question, well, what does Darwinian evolution say about the origin of life? This is important because many people are under the impression that Darwinian evolution explains not only the, I guess, vast diversity of biological life we see on the Earth today, but the origin of life as well. Well, let's just take a quick look at that. Now, we won't spend too much time on this because I think most of you have at one time or another. Have you seen presentations on Darwinian evolution? Just the basics of it, or maybe
A long time
Nova
ago.
or television, something like that.
I had a teacher that knew it very well and a book that knew it very well.
That's right. Yeah, sure. If you've most high school biology classes will have gone over it at some point. Certainly in college, you will have seen it. But so what we're going to do is just look at very quickly. There's lots of ways to describe Darwinian evolution, but the couple of things I'm going to put up here tonight fit in nicely with our presentation tonight, and they're not really argued. The first thing you have to have is self-replicating molecules. It's, we talk about inheritance of certain genes and so on and so forth, and what does it all require? It requires beings that can reproduce, so that requires self-replicating molecules. And the idea is that these molecules undergo random genetic changes, could be from many different sources. Yeah, we could name a whole lot right now if we wanted to. But in any case, they undergo these changes, and they're typically described as very, very small changes. But what they do is they result in some change in the offspring of a particular living creature. And at that point, if those have some beneficial impact on the individual, then nature will select that individual to live and reproduce. So, you know, if you were a dog and you were born with only 2 legs, you're probably not going to make it very well in the world. So the idea that kind of genetic change would be a very negative one, and that dog would not live to reproduce. And the last thing you need, because the changes are considered to be so small, assumed to be so small, that it takes a long time to turn a dinosaur into a bird. And you guys have probably heard that before. Many biologists today believe that birds of today are descended from the dinosaurs in the past. But that required a very, very long period of time, because each of these changes can't be drastic, because the changes, any change that was drastic would likely work to the detriment of the individual. So, let's see here. Now, on this long periods of time, one thing that we're going to touch on in a couple of places here tonight is that even though long periods of time are assumed to be required by Darwinian evolution, the fact of the matter is life appeared on the Earth about absolutely as soon as the Earth was able to sustain life. There's a great deal of evidence today which points to that, and that's really not disputed very much. So what does Darwinian evolution have to say about the origin of life? Well, you can see that without self-replicating molecules, in other words, life, Darwinian evolution has nothing to operate on. There is no way that you can apply Darwinian evolution, at least in this sense, to the origin of life, because there's no living creature for changes to occur to and then to be passed on to offspring. So Darwin himself recognized this, and so when he wrote Origin of the Species, at the very end he speculated a little bit about the origin of life, and he basically said, well, I assume that it was something may have happened in a warm little pond somewhere. So if you hear that terminology, warm little pond, that came from Darwin. When chemicals just sort of came together and formed the first living cells. Now you will see that Darwinian language is used a lot when it comes to origin of life pathways. By pathways we mean chemicals combining in ever-increasing complexity. See, there you go, there's a Darwinian term right there, ever-increasing complexity to form the first living cells. You'll see survival of the fittest. And in some cases this kind of terminology has some legitimacy. But we just want to make sure that you understand that Darwinian evolution does not explain the origin of life. It cannot. There's no mechanism. Okay, so what we are going to do tonight is discuss a characteristic of the most basic building blocks of life that Darwin in his day had no idea about. And this characteristic is much simpler, actually, than the simplest living cell. Yet it's crucial to the understanding of even how the most basic proteins that get incorporated into living creatures assembled in the first place. Now, before we do that, we're going to go back. See, I was going to use this illustration, but I thought, oh, you know what? Nobody's going to understand this because I'm going to be the only one old enough to remember it in my presentation. But then I saw there's a movie coming out on it. That's Mr. Peabody. Have you ever seen the commercials? Mr. Peabody is coming to the big screen. So I guess I do get to use it. We're going to get it to go with Mr. Peabody and Sherman in our Wayback Machine about 60 years. I'm so looking forward to that movie. Okay, Stanley Miller, Harold Urey, University of Chicago, 1953. Probably the most famous, or at least well-known, experiment performed in the entire field of biochemistry. Not necessarily the most important one, but it's certainly the most well-known. Now, his work, Miller, by the way, was a student. Harold Urey was his instructor. There's a real interesting story behind this all, how this all happened. I wish I had time to go into it, but I don't. You ask me afterwards, I can tell you about it. But in any case, Stanley Miller wanted to do this for his doctorate. And he had this idea of how from non-living chemistry, non-living molecules, that we might form the precursors to life. Those being amino acids. So he thought that the ideas he was basing his work on was a hypothesis put forward in the 1920s by Russian biochemist Alexander Oparin and British geneticist John Haldane. Now, what they both hypothesized is it had to do with the atmosphere that made up the very early Earth. And we're talking about early Earth, we're talking about 3.9, roughly, 4 billion years ago. Oparin and Haldane looked at the outer planets, the gas giants that are in our own solar system, Jupiter, Neptune, Saturn, and they looked at the atmosphere. Even in those at that time, they were able to tell something about the constituents of those atmosphere, and they decided that, well, you know, when Earth was very, very young and primitive, it probably looked like one of the gas giants in terms of the atmosphere that were there. So they said the early Earth must have been made up of, you don't have to remember these, you will not be tested later. This is made up of methane, ammonia, water, and hydrogen. Now, this is I think on your, this is on your little cheat sheet with your definitions. This is what would be called a reducing atmosphere. The chemistry term, a reducing atmosphere. Now, the simple way to put that is, what that means is it efficiently removes free oxygen from the environment. Okay, what's the deal with oxygen? By the way, life could never originate in the atmosphere that Earth has right now. It sounds kind of counterintuitive, doesn't it? Could never survive. The reason is because we are breathing right now about 20% oxygen. Is anybody familiar with how hydrogen peroxide works?
It makes your hair a lot lighter.
It makes your hair a lot lighter. That's exactly where I was going with that. There's always one. I didn't think of that, actually.
Makes blood foam.
It makes blood foam. All right.
Yes.
Well, what it does is, hydrogen peroxide is the chemical formula for it. It's H2O2, which sounds kind of familiar. It's kind of like H2O, except there's another O in there. Well, as it turns out, when that molecule hits the hemoglobin in your blood, the part that makes your blood red, the iron, that single molecule of oxygen breaks off from H2O2. Now you have what's called monatomic oxygen, which is an extreme oxidizer. What that means is any bacteria that's in that wound, actually anything living, gets oxidized, which is another way of saying killed when it comes to living tissue. And that's what you want. And if a little of your own tissue gets killed at the same time, that's all right, because you got a lot where that came from. But there's only a few bacteria in there, relatively speaking. So that oxygen oxidizes that living material, that bacteria, And that's what helps, heal your wound. So, you can't have an atmosphere where things get oxidized, especially if you're trying to build something up from basic chemistry to something that's very complex, like a living cell. A living cell is extremely complex. So you can't have a bunch of oxygen around. So you need something that when oxygen is produced, and it is produced in the early Earth, not in large quantities, but in large enough that it's a problem. You need something for it to combine with so it doesn't bother these little life molecules that are trying to get started in our Darwin's warm little pond. So it was necessary to have a reducing atmosphere. And that's, of course, what O'Parent and Haldane hypothesized. And it was what they call strongly reducing, so it very efficiently removed oxygen. Now, in their idea, in their hypothesis, they said, okay, at that point, these amino acids which form, and amino acids are what you refer to in various ways as precursors to life, prebiotic life. There's one slipping my mind, but in any case, they're the simplest things that go into building life molecules, proteins. We're going to see that a little bit later, how amino acids assemble into proteins. Or at least in a simplified way, we're going to say it. So, and their idea is that these amino acids can then peacefully assemble and get more and more complex as time went along. And they didn't need existing biological systems. And why do I say that? Because right now your body's producing amino acids by the boatload full. They are absolutely necessary to the construction of proteins, and proteins is how you stay alive. The vast majority of amino acids being produced on this earth right now are produced by living systems, whether it's you or a tree. It doesn't matter. But before life is found on the earth, then you have to have some more basic system there to produce these. Now at that point, O'Parent and Haldane said, well, they just sort of began to self-assemble to the self-replicating stage. Now, in the 1920s, when they came up with this idea, in the world of biochemistry, science is moving astoundingly fast. I did this presentation 4 years ago, and there's a lot more happening now. There's a lot more known now than there was even 4 years ago. In the 1920s, nobody had a clue as to how complex cells were. So it's not, I wouldn't hold anything against these guys by they're doing a lot of hand waving down here because they really didn't know how it happened. They just had an idea that this is what had to happen, so that's what they put in their proposal. Now, as a point of reference, there are about 600 different amino acids in the simplest human protein. So the simplest protein in your body right now has about 600 amino acids in it. These are simple molecules. You'll see it in a minute. And your body contains about 100,000 different types of proteins. That's a lot. As a kind of an aside, if one of those 100,000 proteins, a particular one, ends up being malformed in a certain way, you end up with mad cow disease. A single protein. That's important. Now we're going to spend a few minutes on this very famous Miller-Urey experiment because it seemed to confirm this hypothesis up here. And that's what all the excitement was about. And that's how the experimental basis for which we get to the very widespread acceptance of you've heard the term primordial soup, from whence all life arose. Okay, well that's kind of where that came from, is this machine that Stanley Miller built. That's just kind of a stylized version of it, by the way, up there, so you stay behind it and look really cool. This is what more like the real laboratory instrument looks like down here. It's rather more complicated and more sciencey. Anyway, So what happens in this apparatus is real simple. And I'll use this as a better illustration because it's just easier to see. What you do, you take this glass globe here and you remove all the air from it. And you introduce this atmosphere right here that O'Perrin and Haldane came up with. You put those constituents inside here. And then you take something up here for all you Saturday mechanics that looks like an old type car ignition coil up here, and that's actually what that is. I don't know if it's in an automobile, but that's an ignition coil. And it creates a very high voltage. And you see there's a little electrode sticking out in here, and another one right here. And they cause the arc to jump across between these two electrodes, so you have bzzz going on in this globe. And that energy input is what catalyzes, what causes these reactions to take place between the methane, ammonia, water, and hydrogen. Okay, so that's kind of how it works. Now, what are the results of this that have everybody going from the 1950s up until maybe the early 2000s? Miller's apparatus produced, right now, you're made up of about 20 different amino acids. Matter of fact, there are 20. You also have other stuff too, sugars, but we're mostly going to talk about amino acids. And what this device did, see if you look right over here, look at this top globe right here, it's kind of hard to see, sorry about that, but it's kind of this brown sludgy material that's up in here. Well, if you take and analyze that, most of it's kind of a tarry substance, which is not very useful. Most of it's kind of a tarry substance, but you can pick off and analyze the other constituents that you get up there, and in fact, you produce many of the amino acids of the 20 that you find in biological systems. Most of them in small amounts, but they're there. So, so for years, this experiment was hailed as virtual proof that the primordial soup that Darwin referred to could have eventually produced self-replicating molecules. Now, as is often the case when it comes to scientific studies, what you read in the popular press and what the actual scientists say about it are sometimes quite different. Miller was much more modest himself, and all he said was that, well, the experiment appeared to show that synthesis of some prebiotic, and that means just pre-life molecules, is possible from non-living materials under the right conditions, these being laboratory conditions. So that's all Miller said, and I mean, that was a lot, that was saying a lot. But it seemed to confirm this. So all you really needed was a spark discharge, you had this assumed atmosphere, and all of a sudden you have this prebiotic or primordial soup that people talk about from which life sprang. Now there's a couple of problems with Miller's experiments, which we know today, and again, which are not controversial. And the first problem is based on a lot of geophysical evidence that was gathered since Miller's experiment took place. And as it turns out, geophysicists now understand that the Earth was really, this did not exist. The atmosphere of the early Earth was mostly determined by volcanism, volcanoes, and the gases that got spewed out of those. And so it doesn't, when you think about it, trying to compare the Earth to Jupiter or one of the outer planets, you know, that's got to be kind of iffy to begin with. And geophysicists tell us now that no, the atmosphere really wasn't much of a reducing atmosphere if it was at all. So the atmosphere is all wrong. And when you put in the atmosphere inside this globe that scientists now believe covered the early Earth, you get virtually nothing out of it. You get some, but you don't get very much. Now, Miller's work is still hailed as a great advance in our understanding of how prebiotic molecules might form, but it's now considered to be irrelevant in the fields of biochemistry, which is a big, big deal. When I say that's not me using the word irrelevant, that was the word that was used by one of the scientists at the last Origin of Life conference, I think in 1999, with poor Stanley Miller dying of cancer in the audience. And yet his experiment was termed to be the great experiment, but irrelevant. So now the second problem with Miller's apparatus is by far the more difficult one even. But before we go there, we're going to make yet another short slide trip to explain a popular and current theory proposing the direct introduction of prebiotic or even living molecules on Earth. Oh, okay, that's just the one of the points I was trying to make earlier. Looking somewhere else. I don't mean this, by the way, to be a stab at anybody that believes this. It's just kind of, I thought, well, that'd be kind of cool. You see, there's a website up here. Kind of an interesting term, huh? Okay, now, given the random and almost infinite ways that, and we'll see this a little bit later, that amino acids can combine with one another, and they readily do combine with one another. So you have these 20 that make up your system right now, your body. If you took a bowl of all 20 of those and mixed them together, you would find that they would stick together very well. But they stick together in kind of but they do stick together in random patterns. So the idea is that enormous amounts of time are required to hit on the right combinations required for even the most primitive forms of life. Now the problem that follows on from that is simply this, that remains of photosynthetic life have been discovered in rock formations dating to about 3.8 billion years ago. Now, that's a long time ago, but the important fact, though, is that that is the earliest possible moment in Earth's history where biological life could endure the conditions there. We've heard of different eras in the age of the Earth, referring to the age of the Earth. The era that was geologists used before life was called the Hadean era, Hadean, which would be what do you think that referred to?
Hell.
Yeah, because the conditions there were hellish in terms of volcanism. The crust of the Earth was molten from at some point or points. So there's no conceivable way that life could have existed there. And yet in a very, very, very short period of time, geologically speaking, we go from those conditions to life. Well, how did that happen? How could it possibly have happened so quickly? This poses a real problem for origin of life researchers. So, because of the difficulty in identifying these pathways to life that could occur over such a long period, or such a short period of time, we've ended up with a group of serious adherents to an idea called Panspermia. So, as the name suggests, the idea is that the Earth was seeded with life from somewhere or many places out there. This is rather amazing that so many origin of life researchers now think that life did not and could not have occurred or happened on Earth. Life must have come from somewhere else and been transported here. Now, some of these prebiotic molecules, again, I'm talking about amino acids, have been discovered in meteorites, comets, even in deep space. So the theory goes that if these molecules didn't originate here on Earth, they must have come from one of these other places. Now, these ideas are very speculative, and there's a debating term called begging the question that applies here. They don't answer the question as to life's origin. They just move the problem somewhere else. What do we mean by that? Well, okay, if life didn't originate here, but somewhere else in the universe, the obvious question is, well, how did it originate there? So the mere fact that you could explain how life got on Earth from somewhere else doesn't explain how life began. It just moves the problem somewhere else. But it's important, I think, we address this because there really is if you follow this kind of thing at all in in, 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 There's just kind of steady drip of articles in the popular press whenever some scientific discovery is made of a discovery of prebiotic molecules in space. We're going to talk about a couple of those here in a moment. So that's why I think it's important to address it. Okay, meteorites from Mars. These aren't jokes. You know, this is serious science. And there's a lot behind this. The rovers on Mars. See, this is what's cool, because when I did this 4 years ago, there were no rovers on Mars. Not at all. Now we got all this interesting data from the rovers on Mars. So they've given us significant evidence that there was, at least for some time in some places, running water in the warmer atmosphere on Mars, long before those conditions existed here on Earth. So the idea here is that life began on Mars. Mars was impacted by asteroids, which basically blew big holes in the surface, some of which could have contained these living molecules, or at least amino acids. Those got transported to Earth, and that's how Earth ended up with life. So that's one idea. The next idea, and we have now actually discovered amino acids existing in comets. Not very many, but there are some that are there. So the idea is that, well, comets brought, and most scientists think comets brought a lot of water to the Earth early in its history. So the idea is, well, why not? Could they have not brought prebiotic molecules as well? Here's a picture of, I don't have a scale there on that, but that's a fragment of what's called a Murchison meteorite. It's very famous. Found near the town of Murchison, Australia in 1969. And what's interesting is when they drilled into the center of it, brought out some, what they consider to be pristine material that is not degraded by Earth's atmosphere or otherwise invaded by, it's hard to look at anything here on Earth. Life is everywhere. I mean, it's everywhere. So whenever you bring something in from outer space, it's very difficult to tell whether what you see in there, if it appears to be something like life. By that, I don't mean living molecules, I mean amino acids, precursors to life. It's hard to know whether they came from Earth or where the meteor hit. So this is a serious problem with NASA, that they take great pains to try to get around. But by drilling into this rock, they pulled out some material from the middle of it, which they consider to be pristine, and there were some prebiotic molecules in there. Okay, who believes in this Panspermia thing? Well, let's try theoretical physicist Stephen Hawking, who's talked about it. Let's talk about evolutionary biologist Richard Dawkins, very well-known name, many other naturalistic thinkers. So they think that this could have very easily, these molecules could very easily have come even from interstellar space and landed here on Earth. And that's how they got their quick start here. Now, what would cause a group of highly regarded scientists to come to these kind of conclusions? I mean, it really does seem really very much like science fiction. Well, leaving aside the possibility that life arrived on Earth as complete organisms, in other words, aliens, that's another set of aliens, which is a subject for another time. And by the way, I'm not touching on it because there's no evidence. I mean, outside of crop circles.
What was that?
Outside of crop circles.
Oh, I see.
No, there is no hard scientific evidence that we have been visited, and especially the Earth was seeded by aliens. There is zero evidence of that, so we're not considering it. But leaving aside that possibility, and returning to our major theme for tonight, there's a characteristic that most amino acids have that makes them either suitable for building life molecules or quite simply makes them completely unsuitable. And this is where we get to our main topic. chirality or chirality. So if I pronounce it with CH with a ch tonight, you'll understand, because I keep wanting to do that. It comes from the word chiral or chiral, derived from the Greek word for hand. Okay, well, why would we call this characteristic of amino acids chiral? Well, they are compounds based on carbon, nitrogen, oxygen, and hydrogen, which we have a lot of here on Earth. And this is, again, a characteristic of amino acids. Now, here we go. Here's an amino acid. This is one of the simpler ones, not the simplest ones, but this is called alanine. Alanine is very common. Again, your body's producing it right now, but both of us, well, because it's needed in a great many of life processes. Virtually all proteins would have alanine as an amino acid in them. But what happens is when you, in what we call abiotic systems, that is systems that don't involve biology of something that's living, these are found in nature. Even though they're the same molecule, they're found in two different versions, a right-handed version and a left-handed version. They are the same chemically for most purposes, but like your hands, you have two hands that are the same, but they're not superimposable. They're both hands, but they're not superimposable. Neither of those molecules. But they have the same atoms in them, the same number of atoms. They're just mirror images of one another. Now, where I lost my place here. Okay. Now, if we were to produce a batch of those right now using, other than very sophisticated equipment, you'd find that you would get a mixture of the two of these. When I say the two of them, I mean the right-handed and the left-handed version. By the way, each one of those by itself, and here's our next big word alert, is called an enantiomer. Enantiomer, and that's on your list. It's on your yellow sheet. And that mixture, and there would be produced as a 50-50 kind of mixture, and that kind of mixture is a medical and biological term called racemic, which is 50-50. It's a racemic mixture. So you have as many right-handed molecules as you do left-handed. So we have three important words. We have chiral, which is the characteristic of handedness or mirror image. We have enantiomer, which means each separate hand or image. In racemic, which is a 50-50 mixture. And we come now to our fourth, which is homochiral, which is a collection of a single enantiomer. In other words, if you had a right hand, you only have right-handed versions of that molecule, or you only have left-handed versions of it. That would be a homochiral collection. Now, the Stanley Miller apparatus produced these molecules actually fairly easily. And what you find out when you analyze the results of his experiment and the amino acids that it produced is they were always of a racemic nature. You had equal proportions of the right-handed and the left-handed molecules. You're going to see in a minute why this all sounds like, well, so what? But you'll see in a minute why that's so important. In fact, there it is. Okay. Homochiral sources of amino acids are absolutely required in order to produce useful enzymes or proteins in your body or in any other living creature. They cannot be produced with mixtures. They have to be. They could be right-handed, they could be left-handed. As it turns out, in your body, mine, every creature on Earth, every living system on Earth is made up of left-handed amino acids and right-handed sugars. There's no debate about this in the scientific community. when any of the amino acids, if in one of your cells, somehow a right-handed amino acid was used to build a protein, that protein would be malformed. You would be in big trouble. This is interesting, we've got this right, remember? Same chemical makeup, both of them. But when they're in living systems, and you'll see a little bit later why this is. What happens when you have a malformed protein? Just one?
Things don't work.
I know, that was working earlier. I don't know why it's not working now.
It breaks your PowerPoint.
Let me see where my, where's the mouse for this?
Is that it?
Yeah, use my mouse clicker. Anybody, has anyone seen these pictures before or ones like them? Is that familiar, these children? 1960s? Thalidomide. What is that? Thalidomide. Thalidomide was a drug produced in Europe, and it came out, it was marketed for use by women who were undergoing morning sickness while they were pregnant, in the early stages of pregnancy. And they were prescribed thalidomide. And thalidomide was made up, amongst many things, other things, was made up of amino acids. And as it turns out, the amino acids that went into the drug was a racemic mixture of right and left-handed molecules. Well, at that early stage in a child's development, in the development of the fetus, the introduction of right-handed amino acids into their system, they had no defenses against it. So again, you have just a single malformed protein that went into building most of the hands and feet. And because of that malformed protein, they ended up with more like, they have flippers, that's what their hands and feet look like. So this is what the introduction of other than homochiral amino acids into your system, with the kinds of things that can happen. There's about 3,000 of these children still alive in the world today. Actually, most of them are doing better than you might think.
Fred, what effect would it have on adults? The wrong-sided amino acids?
This is actually a debated question today. There are some doctors, researchers are proposing that all drugs given to adults be made up of homochiral selections of amino acids. Your body does have defenses. Not all the, in fact, some of it just gets ignored. Remember, the production of these amino acids, the incorporation of amino acids within your body takes place within the cell itself. And the cell itself has defense mechanisms to keep the wrong stuff from getting in and being incorporated. Also, the amino acids that are being, many of the amino acids are actually, you don't get from the external environment. You've heard the word essential acids, non-essential. Your body produces about half the amino acids that it needs by itself, within the cell. And those are all made by a blueprint, via a blueprint called DNA. And your DNA only makes left-handed alanine and amino acids.
Is that why the amino acid supplement I buy, it'll have a L? Is that meaning the left side?
Yeah, actually, that's that's getting to be an old term, but yeah, the enantiomers, the left and right-hand versions, sometimes in the scientific literature go by which words, L for left and, no, not R for right, D for right.
Oh, no.
It stands, it's for dextro something, which has to do with what you identified chemically. Yeah, they have other terms for them, they use now R and S or something like that. I can't remember what they are. So, but many researchers now are saying that that drugs that are delivered via amino acids should all be homochiral. Now, And there are ways of producing them, homochiral selections of molecules outside of living tissue, but they themselves involve already existing homochiral collections of molecules. So it's a very expensive proposition producing these, considering how ubiquitous they are on Earth. I mean, like I say, every plant you come across. By the way, the plants make all the amino acids they need. You don't. That's why you have to eat plants. So you have to eat peas. Because you don't make all the amino acids that you need.
So just so I'm following, and is that it always has to be left-handed with the sugar?
Right-handed for sugar,
Right-handed for sugar, and then left-handed?
for the amino acids, or for the, for amino acids is left-handed, for sugars is right-handed.
I see. And the experiment produced both?
Yeah, it only produced amino acids, but of those, it produced both left-handed and right-handed versions equally.
I see, okay, thanks.
That's why it could never end up as a living tissue. But
Gotcha.
you're gonna see an illustration of that here in just a couple of minutes. Okay. Now, okay, so we know that we need left-handed molecules for living systems all throughout Earth. Now, you could build living systems on all right-handed if you wanted to. I mean, there's no reason, there's no preference to the right or left. But once you get one established, it's got to stay that way. You can't mix it. You can't ever mix the two. So, so the question remains on early Earth. Let's go back to the early Earth. Well, if you're going to be assembling amino acids into these long chains, which are eventually going to become proteins, which are going to go into the cellular structure of a living creature, even if it's a bacterium, what do you do when you've got all this? When you've got this little primordial soup or ocean or pond or wherever it is, it's full of what has to be a racemic mixture of the two. What do? You can only make these make them correctly out of everything has to be one enantiomer, either the right or the left. So the origin of life researchers, the questions become, well, okay, we needed all these left-handed amino acids. How do we end up with just left-handed amino acids in our warm little pond? Can that happen? And the answer is kind of yes, sort of. Let's go back. These poor kids, I hate looking at those pictures. Okay, yeah, even small amounts of the opposite enantiomer is destructive to the life process. You end up with malformed proteins. Okay. Oh, yeah, here we go. In nature, homochiral molecules are produced exclusively by living systems. If you find a collection of left-handed amino acids somewhere in a little jar, you'd know that a living system produced that. It can be produced with very specialized equipment in laboratories, and they are. But again, it's a highly complex procedure which requires to do that. You just don't find them laying around. Now, so this is just a little diagram of a cell, which, boy, I wish we could have a time for an hour of just talking about a cell. It's fascinating. But what this cell produces is, and what you can see here is, these are all left-handed versions of this particular amino acid. Well, actually, it's not. That's just kind of a generic diagram of an amino acid here.
What was the handedness of the Murchison amino acid?
Oh, well, interesting question. They found big word alert, enantiomeric excess. What that means is now you have a collection where there's a slight difference between the number of right and left-handed molecules. Well, not necessarily slight. There just has to be an imbalance. Both are there, but there's an imbalance.
Is that significant?
And they found when they opened up the Murchison meteorite, there's a slight enantiomeric excess of some of the amino acids in there. And of course, I got people really excited. Because what it said is, whatever incorporated those amino acids into that meteorite, there was a selection process going on somewhere, somehow, that caused there to be more of the one-handed than there was the other.
It wasn't just random.
But they
No.
also didn't have any way of knowing absolutely that they had the entire meteorite, because how much is destroyed coming through the interval?
Well, you have to realize that, you know, of course, amino acids just being a molecule with a few atoms on it, you know, there's, you know, even a very small sample might contain hundreds of thousands, if not millions, of a molecule out of the meteorite. So if you look at a tiny bit of that and you found it to be 90% left-handed amino
I
acid,
probably
that would be
know
very
it.
significant.
Yeah.
Because you wouldn't find like one half of the meteor, the right half would be right-handed and the left-handed. It wouldn't work that way.
Yeah.
They would tend to be mixed. Yeah, not like buttermilk, kind of.
But you said there's a slight imbalance, so I'm thinking, well, what if you had the other half of the intermediate, would it theoretically balance out? But
I don't think so.
not
No, I don't
necessarily.
think so. And they probably took several samples from several different places in the same meteorite, got the same result. I'm assuming that, I have not read that study. Scientific studies like that are actually hard to come by, you gotta pay for them. They're like really expensive, and then why would you spend $35 on an article you can't understand? Now, these guys get pretty serious about their work, so some of this stuff is. So I'm like most people, I depend on other scientists who are good at explaining things to the rest of us for a lot of the information that I get. We're pretty much all forced to do that unless we're biochemists. Okay, however, if you were Dr. Fu Manchu right over here, that's another, you have to be old enough to remember Fu Manchu. Well, he's black and white, come on. Yeah. So only in the 1930s could you have a Chinese villain named Dr. Fu Manchu, played by Boris Karloff. It's not like there weren't Chinese people around then. I don't know why they did it. Maybe it's just because it looked better on the marquee of the theater. I don't know. But anyway, if Dr. Fu Manchu were making up in his little beaker there, a bunch of alanine or whatever, he would end up with a racemic mixture of molecules. So it's only living systems that we know of produce these exclusively one-handed collections of molecules. Okay. Okay, let's move on. So, how did you explain the existence of homochirality? How did you end up with these selections of one-handed molecules? And scientists have done a lot of work on this. And they really have made a lot of discoveries. Some of them are very, very interesting. Now, I'm not going to spend very long going over these just in the interest of time. These are like things that if you wanted to, you could turn into a presentation in its own right. So I'm necessarily using a lot of shorthand up here. I hope you understand that. And some simplifications. Not all amino acids are chiral in nature. Stuff like that. As it turns out, and by the way, this is just to get your attention. Okay, you will be tested on this. This is a representation of a circularly polarized ultraviolet light. Okay, so whoa, what is that? Well, of course, the ultraviolet part of the spectrum is just above. If you go to the blue end of the spectrum, you've seen a rainbow. You go through all the colors, and the last one's is blue, the end is violet. And if your eyes could see any further, then the next one would be ultraviolet. Okay, that would be the frequency of the light. So people have determined that at certain frequencies of ultraviolet light, at certain intensities, if you aim it at a beaker full of this racemic mixture of amino acids, it will selectively destroy one of the enantiomers, so you could end up with more of one hand than the other hand. So they say, well, maybe that's what happened. Maybe this all happened in outer space. And then once this ultraviolet light did its work, these amino acids were incorporated into meteors and found their way to Earth. Scientists have discovered that certain very common materials, like this is a crystal of what's called calcite. It works kind of like a activated carbon filter when you put it in your water faucet and it gets all the chlorine out of your water. What happens is the chlorine tends to stick to the carbon, the activated carbon. So and that's why the filter eventually gets plugged up. It doesn't work anymore because there's no more surface area left for the little chlorine molecules to latch on. Well, these chiral molecules on one face of the crystal will tend to collect the right-handed version, and another face of the crystal will tend to collect the left-handed version, and that's how they all got separated in the early Earth. That's what people hypothesize. In fact, this particular idea was found on the homepage of the American Association of Biology Teachers about 10 years ago, which I'm sorry to this day I did not keep a copy of because it is since gone and you can't find it anymore. This was their claim as to how homochirality came to be on the Earth. They were just giant crystals around at the time. There are a lot of problems with this idea. And it's like, okay, so they're stuck, but at some point they have to get unstuck in order to do their work, and how do you keep them from being mixed together again? And so there's just a tremendous number of issues there. And so at this point, that's really kind of now a scientific curiosity more than anything else. There's some others. I'm not going to go over these because they're too difficult to explain. I think at the time that we have. There are ways that you can amplify a slight excess of one enantiomer chemically, but there's no proof, there's no evidence whatsoever that anything like that was going on the Earth in its early history. And some scientists just say, well, you know, homochirality isn't important at all. You know, life got started some other way, and then homochirality arose out of that. And at that point, the original molecules sort of went away because there's no more need for them. And that's how we get to where we are today. So there's a lot of hand waving again that goes on. And the biggest problem is the scientific evidence they have for a lot of these ideas are things that have been done in the pristine confines of a laboratory experiment with designed molecules done by people with doctorates in biochemistry. Things like this. So, you know, when you try to transfer a lot of this understanding into the real world, when you have a little pond out there which is not clean, it's not the inside of a laboratory anymore. It's the real world, trying to do a chemistry experiment in the dirt. That's kind of what a lot of this is about. It's just a lot of it doesn't transfer over very well. So again, this is an idea that has been put forth with some, I'm not going to say there's no evidence at all, there is some. But most scientists look at these, the origin of life researchers look at these possible pathways today, and they just go down the list. Too many problems, too many problems, too many problems, too many problems. Okay. None of these produce a true homochiral environment. They only amplify it. Okay. So what's going on here with this primordial soup or the warm little pond idea that they had? Okay, let's just assume that we have a method for producing these extremely large quantities. This all turns out to be a probability problem. When you come right down to it, you're talking about molecules randomly assembling. Let's see if I have a statistic. Next page, good. Randomly assembling. So, in a very short period of time, to something that's exceedingly complex, i.e., a cell, even if it's a single-cell organism, like some kind of bacteria. So. But let's just say you have all these left-handed amino acids that are available to you, and they're all sitting there in this nice little pond. Well, you've got to keep them that way. You can't have any of the other-handed enantiomer that somehow gets dropped in there at some point. You only have to have this collection to begin with. You have to keep it isolated for a long, long period of time under perfect conditions. And again, you have to have, trying to do an experiment out there in the dirt, folks. I mean, it's a little different now because you find life everywhere that interferes with these kind of experiments. But even in even just rocks, a rocky environment, you just don't find laboratory conditions out there. So a lot of the ideas that people put forward have only been shown to work in the laboratory with experiments designed by intelligent people. I hope that's not lost on you. And these conditions have to remain static for very, very long periods of time. Let's see why in just a moment. Okay, let's say we've got all this, though. We have all of this, all of these things. I don't want you to be too intimidated by this slide. It's not my slide. That's from Fazil Rana's book, The Cell's Design. This is like the whiz-bang explanation of how proteins are formed. Over here, you have all these amino acids. Now there are 20 that you need. They only show a few of them here, just because it gets too confusing. And as I said before, these things have a tendency to just stick together when they come in contact with one another or get really close. And they tend to stick together in long chains. And there's no preference. Valine, glycine, lysine, alanine, they will all stick to one another equally well. And so when they come together, if you put them in solution, then they tend to start moving together. Well, if they get close enough, they just randomly bump into one another is what happens. And when they do, they stick. But for reasons we don't have time to go into. But they form these long chains. Now, when two of them stick together, I love terms, it's cool. They call it's a peptide. When more than two of them stick together, it's a polypeptide.
Jeez.
Okay? That's cool. So you get these long chains. And by the way, this is not a chain that's long enough to do anything anywhere. You have to get up to around 100 before they're useful for anything. Okay, now, if you take 20 amino acids and they form these random linear structures, polypeptides, and you have a chain that's 100 units long, and by the way, the shortest useful polypeptide chain the shortest one in your body is about 124. But if you get 20, randomly assembling them to a length of 100, how many combinations are there? How many different combinations could you have of 100 using 20 components? The answer is 20 to the 100th power. That's a big number. So 20 with 100 zeros after it. That's how many different combinations there are. And only a few of those, a very, very few, would make anything useful. And that thing that's made that might be useful probably would not be useful unless it was in the presence of a lot of other things that were useful. So you see, the probabilities are not good for life randomly assembling. So, now, if these amino acids are left-handed only, once they form these long chains, once the chain starts to get longer, it assumes one of two forms. Either this kind of a helix structure, not to be confused with DNA. If this was part of a DNA chain, it would be miles long. Either they form this helical structure or they form what they call a pleated sheet. Helical structure You look at your hair, there's some up there somewhere, there's one. Look at it under a powerful enough microscope, you would see these helical structures of amino acids. If you were to look at a strand of silk, you would see these kinds of structures predominating. But in any case, once these are formed, they begin to fold in on themselves. They get long enough, they start to fold. And if you get another one over here that's doing the same thing, and another one over here that's doing the same thing, not the same thing, they fold into different shapes because they're different sequences of amino acids. And you bring all of those three together, and they're just the right type, they stick together. Now you have a useful protein. But again, it's not useful unless there are other proteins around to work with it. So you see, the mathematical probabilities here just start adding up, to the point where you can't get there randomly. Not from the time the Earth was able to sustain life till the time life appeared was anywhere between 100,000 and 10 million years. 10 million years sounds like a long time. It isn't. You're talking about these kind of probabilities. 20 to the 100th power in terms of the number of different ways amino acids can assemble into a polypeptide in 100 unit form. Okay. This is all why that the origin of life, the idea of the origin of life having taken place in the famed primordial soup has really fallen out of favor by many, and perhaps even most scientists. Because to accomplish all this list of requirements, if you start adding up all the things we've talked about, it's just too long. The list is too long. The time is way too short. So, it's just asking too much. Now, let me give you another. The simplest bacterium in existence contains 2,000 different proteins. Now, it doesn't matter if it's you or a bacterium. This process is the same. Different results, obviously. But you figure there has to be 2,000 different proteins that go to make up a single cell bacterium, the simplest one in existence. Now, note I said cell. I didn't say 2,000 different proteins being held in my hand. I said within a cell. Even if you have all these proteins, even if you have all 2,000 for the simplest bacterium, where do you put them? There has to be a cell membrane to locate them all in. Oh, really? Where did that come from? See? Where did that come from? Okay, let me add yet another one. This is my last one. Not only do they have to be in a cell, which we haven't invented yet, they have to be at certain locations within the cell, at certain times within the cell, in order to function properly. So you need all the right components at the right places, at the right times. And in the majority of cases, these complicated components have no other function. They only work together as a group. If you take one of them apart, blood clotting. Probably shouldn't read this. Blood clotting. There's a cascade effect. You think I grew up thinking, what does blood do? Oh, it's dried. My blood dried. Good. I'm not bleeding anywhere. Blood doesn't dry. Blood coagulates. It's a very, very complex. Only understood about the last 15 or 20 years how blood clots. It's a cascade effect of different reactions that go on within the human body or any animal that causes the blood to clot. All of these sequence of events that take place have no other function other than to clot blood. And they all have to happen in the right sequence at just the right way at just the right time. And they have no other function. So it wouldn't do any good to randomly assemble a protein used in one stage of blood clotting if the other stages didn't already exist. Oh, okay, where'd they come from? Because they can't be used without the one we just went through. So, see, this problem becomes just about intractable. All right. I'm going to skip our summary here because I think we've gone over this enough. I'm just going to do my little concluding remarks here. Okay. I have to turn the light off for this. This is too beautiful a picture. Anybody seen this before, this painting? Okay. So homochirality is a major problem for naturalistic or mechanistic origin of life theories, but it's only one of great many. So how did life happen? Was it just blind luck through random association of these amino acids, other prebiotic molecules that we somehow ended up with a living cell? Well, a lot of people, among them many scientists, say, yes, it was luck. Yes, it was luck. And the ultimate proof of it is simply this. We're here. Anthropic principle. Yeah. Well, they say that it must have happened through naturalistic processes because we're here, aren't we? So it may be mysterious, but it happened. Now, it's all circular reasoning. All of a sudden you have a scientist making not a scientific statement, but a philosophical one. And that's something you have to really watch out for, by the way. So, now, without serious objection from the scientific community, life at least appears to have been designed. Francis Crick, a word that's familiar to some of you, wrote in his book, scientist, writer, very popular writer, What Mad Pursuit? Biologists must constantly keep in mind that what they see was not designed, but rather evolved. Remember what we said about Darwin earlier. Richard Dawkins, famous writer, in his book, The Blind Watchmaker, biology is the study of complicated things that give the appearance of having been designed for a purpose. But of course, in his view, they are not. Now, this painting on the screen is called The Lady of Shalott, and it's hard to read them. The Lady of Shalott by the, from the poem of Handedness, the same name. And it's done by, painted by John Waterhouse, I think in the early 20th century, if I'm not mistaken, late 19th century. And it was, I think what I like about it, it's what's called the pinnacle of pre-Raphaelite art. Now, what do I mean by that, pre-Raphaelite? All it means is before modern art. Raphael was the first painter to do what we popularly call modern art today. This was just before that. And what characterized painting of that era, in the pre-Raphaelite era, was extreme attention to detail. If you want to come up and look at this accurately, the screen has such low resolution and it's not bright. It's hard to see here, but the intricacy here, the detail, you see these couple birds right here, is simply astounding. And what was their idea? Was to mimic real life as perfectly as they possibly could. In every detail. Now, you could have this painting. If you had this painting, you could decide, well, you know what? It's nice. Yeah, it's okay. I'm going to analyze this painting, and you could spend a good portion of your life analyzing the canvas that it's painted on. All right, See, now it's working for you. Analyzing the canvas that it's painted on, you can take a look at the pigments that are used to paint, put it through a mass spectrometer and determine the precise elements that went into making the pigments, and how they ended up, the brightness that they are, the hues that they are. You could produce detailed scientific tests that detail all of that, and very, very much more. But I'm going to ask you a question. When you look at the painting, are you thinking about the atomic structure of each pigment as it gives its color? Okay? I'm guessing not. Clearly, as you view it, you understand it takes on far more meaning than the deconstruction of its mechanical components. They are, and then we look at ourselves. Are we built as complex biological computing machines, or that have no meaning and no purpose? So why am I here tonight? If I'm a machine, just a complex biological computing machine, why am I here tonight? But why are you here tonight? See, against every instinct within a science from an atheistic perspective tells us that that's precisely what we are. You are a machine. You're a complicated machine. You're a smart machine, but you're a machine. Sophisticated, but still machines. Now, does what I've shown you tonight prove that we're not machines? Well, no, not really. But as we appear ever more detailed into the complexity, the basic structure of life, you have to marvel at the complexity and apparent purpose, okay, apparent purpose, I would say purpose, behind each particular part. So when you couple that with the startling appearance of design and purpose with the another big word alert, the existential nature of human life, what we call the experience of existence. Our experience of the existence of our lives. And you sense that when you see a beautiful piece of art like this. You see what cries out to you. It's that life is more than chemistry. It's more than knowing that a particular sequence of neurons must have been electrically simulated or stimulated in order that we experience these things. So again, referencing the painting, even the word beauty, where does that word come from? What does that word mean to a complex computing machine that just has neurons firing in it in a particular sequence? What does that mean? See, but what's within us screams that we're more than that, and more than programmed by nature simply to continue our existence and ensure the genetic inheritance that we possess. See, those people that are involved in RTV and many others are inclined to believe that a creator of life exists, not in spite of scientific evidence to the contrary, but because we believe that the same, just remember what I talked about earlier, scientific data. Don't often argue about it. We do argue a lot about the conclusions to be drawn from it. And what I'm saying is that Christians will come to the conclusion using the same data that shows compelling evidence that a creator does exist. Now, confronted with this, I've read and heard scientists say this. Oh, okay. But you realize that if we accept the idea that this existence is not all there is, that there might be a God out there somewhere, if we accept that something might have reached into this universe and monkeyed with the system somehow, scientific inquiry will come to a halt. People will simply assume that, well, God did it whenever they come up against a tough problem. And so people will stop investing in science. They'll stop, which is their career, of course. And that bothers them. Frankly, it would bother me too, if I believed that. But what I'm going to say is that such a belief ignores a clear lesson from history, and that is that religious men and women were and are prominent in the sciences And they've made very many, a great many important and really foundational discoveries in science. And they continue to work precisely because they believe their lives are not ultimately determined by simple cause and effect in a closed system. This causes this, and this causes this inevitably. They work because they believe that a rational mind lies behind creation, giving meaning to the creation and to them and us as individuals. Part of their job is to come closer to that mind, partly through their discoveries. Well, I don't really have anything else to say tonight. Did I prove that God did it? Well, no, not really. I hope we got to the point where we realized that to come to the conclusion that it was blind luck, that the reason we're here is asking, and again, based on scientific discoveries that we probably most everyone would agree with, he's just asking too much. I think the evidence is there, that there is a creator, and that we're not here by accident, and that everything in us screams that we're not here by accident. So I hope this has helped you a little bit when it comes to a rather esoteric field biochemistry to show that that's the case. So we have a few minutes for questions.
So, have people actually somehow created proteins where they substituted one R amino acid in the protein and fed it to somebody or something? I mean, is that true?
Well, yeah, again, your body has defenses against some such things. However, mad cow is transmitted through a malformed protein that originated in cattle.
But is it an R type?
I don't know that it's malformed because of a chirality issue or not. That's a good question.
You know what this subject makes me wonder is why the whole topic of chirality doesn't come up more in
Sunday school?
I don't know about Sunday school necessarily.
Well,
But let's just say, I mean, for someone that obviously is well versed on it would be Fres Rana, just to use the group of people that we know well. I've never heard him talk about chirality.
well, he has written numerous papers on it, and in the 3 books, which I forgot to bring with me, it's dealt with in all 3.
Does he consider it just another strand of evidence that leads toward design, essentially? That's his conclusion, which would fall in. I guess that I'm thinking that this is a significant leg in terms of apologetic leg, just like the Cambrian explosion.
Well,
is,
you know,
and
it
some
is,
other-
but this isn't something that's easy to bring up in a casual conversation.
Yeah.
I mean, I recognize that. I spent probably 8-9 months in my original presentation, and then I probably spent a good solid 6 to 8 weeks
But
when I thought about doing this again.
it seems reasonable to me to conclude, knowing of this subject only from you, by the way, thank you very much for your job. What was the point I was gonna make? But at any rate, it just seems significant to me that a paradigm that requires random mutations to be the driving force in a cause and effect, undirected manner for its purposes, which obviously I'm talking about Darwinism, it just seems like this would be a pretty big arrow in that.
Well, it is, and that's why the Panspermia thing is so prevalent now in scientific circles. Because there's a dawning recognition that the numbers are against it, or against the origin of life occurring the way it's traditionally been thought since Darwin.
And mainly because 13.8 billion years just isn't a long enough period.
Well, 13.8, that would be the
The
universe.
universe in
Okay,
4
but so the
billion
idea
per
is
year.
that there wasn't enough time on Earth.
Yeah,
But if you look at the length of time available to the universe,
but even there,
see,
it's not enough.
then all of a sudden you have a lot more time for these things to happen. That's the idea behind it. But when you think of it, let's not forget that the history of the universe is not a static one. Whereas the universe as it exists now looked nothing like this 13 billion years ago, or even 10 billion years ago. And you could make a legitimate article that it was far more inhospitable, the universe as a whole.
Yeah,
It was far more inhospitable to life at that time than it is right now.
sure.
So has someone, oh,
Okay.
did I talk about something? So are we, I'm a non-science person, do we know how old the Earth is for sure? Is it sort of all scientists agree from, lots of scientific data that it's definitely this old? Do we know that?
Well, okay, let me see, I'm gonna phrase this properly.
Okay.
Hugh Ross says that he has never met a scientist, and I'm sure he's met a lot, who believes the Earth. Okay, the scientific figure you'd read in any textbook, normal textbook, would be what, 4.5? 4.2 billion years old. Okay, that's the standard answer. There are obviously some people who believe the Earth is 6,000 years old. But outside of a scientist who already believes that to be the case because they think a particular interpretation of the Bible leads you to that conclusion, no, he's never met one that didn't believe the Earth was 4.2 billion years old. Did that make sense? I might not have said that the best way.
Right, and that's because of a line of evidence from several different disciplines of science kind of emerged.
Yeah.
It's widely accepted.
I see.
It's
Why do we say
Bordering on, the only thing to dispute is whether it's empirical or not, but it's widely supported scientifically.
That's
So
another
keep-
great talk by the way, by Fred Luffman.
What's
Well,
that?
we did do a presentation on the age of the Earth,
Uh,
probably a couple of years ago now. But it was based, so we based the lines of evidence are based on radiometric dating, dendrochronology, which is looking at tree rings. You might wonder how tree rings get involved in this, but they do. Ice cores, and layers that are laid down in lakes and otherwise rather calm bodies of water called varves. So there are several lines of evidence which lead you to believe the Earth is, even if it's not billions of years old, is far older than 6,000 years.
Sure. So given that, has there, and I'm taking a simplistic view of this, is that, has anyone, like mathematically tried to draw that out into equation, like taking, you said something like, it was one number which was like 200 zeros or something like that behind it, or
Yeah, 20 to the 100th power.
20 to the 100th power, and then to have these things form together, that maybe is, 1 out of something billion or
Yeah,
something like
yeah.
that. And then you start putting these together, and then you go, well, let's say that it only took like a minute for this thing to happen. That means that the Earth would have had to have been like 1,200 times long. Has
Yeah.
anybody done that?
Yes. Oh, no, it sounds like a good exercise for the student to me.
Big calculator.
I have actually seen that. Hugh Ross has done that with some scientists in, I want to say, Texas A&M University. They actually
William
worked it
Dembski?
out. What's that?
William Dembski, maybe?
No, it wasn't William Dembski.
Who's also done work on that.
Yeah, he probably has done it too. It's probably been done by multiple people. But if you count the number of seconds, or if you figure out the number of seconds since the beginning of the universe, it's actually not that big of a number compared to the numbers that Fred's talking about.
No, to give you
I forget what it is,
This
but
is
it's
kind of
like
like
10
an
to the
apples and
20th or
oranges thing,
something.
but people have actually calculated the number of atoms in the observable universe. It's about 10 to the 80th, roughly.
Hmm.
10 to the 80th, 10 with 80 zeros after it, the number of atoms in the observable universe. And the probability of one protein by random self-assembling into something that would look useful is 20 to the 100th power.
Because it almost seems like if you weren't, you were gonna take just a complete, idea that there was no grand design or anything like that, you seem like you would very quickly have to come up with that, well, it couldn't have happened on Earth, because, it's by far, it would take a lot longer, the Earth would have had to been a lot. So then you then have to go to, it had to happen in outer space, because that's a lot older.
Well, that's why a lot of, there's so many scientists that are so interested in what's going on Mars right now. They can't wait to get data back that might indicate the presence of life. One thing that would be, if there was evidence of life, by the way, the geochemical evidence that you get for life, you can't determine things like chirality. Because it's too old, the amino acids are long gone. You just have impressions in rocks and so on. So the only thing you can tell is that there is some evidence that life might have been there at one time. Now remember we talked about meteors from Mars?
Right.
Well, gee whiz, you think it's possible you could have meteors from Earth? See, you get into those kinds you could get into those kind of arguments. It's a lot more likely that life here might have been transported to Mars than the reverse, simply because we're a much larger planet. More asteroids hit the Earth, there's more material ejected from the Earth, so you'd expect more meteors from Earth to be found on Mars than the reverse.
Solar wind blows that way too.
Oh,
That's true.
That's interesting.
Also, I mean, if you did find evidence of life on Mars, I'll tell you what would be the kicker, if you found evidence of life on Mars and it was right-handed, the amino acids were right-handed, that would be a big deal. Make you wonder.
That would lead into my question that now seems good, so I'm trying to figure out if you had a rock of some sort, some substance, and it had a life, it had an amino acid that could build some kind of building block of life, and you sent it through outer space. I mean, is there something about that rock that insulates it from all the
Exiting and entering.
Yeah,
Well,
space is a tough place
yeah,
to
it's
live.
kind of like,
It
it's
is.
not inhospitable life in itself.
It's a
you
very
know,
inhospitable
absolute zero
place.
in space, so it's like,
The ionizing radiation,
Ionizing
some of the astronauts, and even in the space station, when there's a solar flare, they have to take refuge in a special part of the
Oh, yeah.
space station to
Somehow-
avoid being.
whatever's transported
Irradiated.
in that rock substance would have to somehow be insulated.
Yeah,
from-
and scientists, they've looked at that, and, the penetration of gamma rays, for example, into rock, and at what point would an amino acid remain intact, or some biological component might remain intact inside. These are all, I mean, they're interesting questions. They don't necessarily prove anything, they kind of give you an idea of what might have happened. See, some scientists say, well, however life got started, the evidence of it is no longer around, so we're kind of out of luck. But they still would insist on there being a naturalistic explanation for life.
Okay. This panspermia idea, what are they actually saying would be transported? Are we talking about amino acids, or are we talking about proteins? Are we talking about amoebas, or what?
Well,
What is
all
it?
the above, it could be amino
Amino
acids, could
Humans?
just simply be amino acids, like Stanley Miller made in his machine, or it could be aliens. I mean, all the way up to and including aliens. I love science fiction.
Yeah.
I mean, I've read several books with that idea.
I just happen to think, we actually had a visitor here, it was probably over a year ago, that was actually worked in the laboratory of the Miller-Urey. They still have the apparatus there or something like that. I can't remember who that was.
That was our one and only real rocket scientist.
Yeah, he was a rocket
He
scientist.
was
right.
an actual
Yes,
rocket
yes.
scientist.
And he actually said that it's this culture that no one dares look outside the box. They got this way of thinking and no one looks outside. He actually would ask around and say, has anybody actually worked out the numbers? Have you ever thought about that? And, no, why would we do that? And so he finally ended up working out the numbers and said, okay, come on guys, this isn't this doesn't make any sense, and then he couldn't even get their attention, I guess.
Well,
Yeah,
I think his point was that a lot of working scientists don't really think the broader questions
Yeah.
through a lot because they're so, really enmeshed in their very specific disciplines within, the research grant or what have you.
Why
It's
don't
a
we,
shame.
if you'll permit me, Wally, why don't we real quick tell people what's up next month?
Oh,
Yeah, can we do that now, Jim?
Right, yep.
Yeah.
Let me